Well, thanks for having me. Love talking about Nomad, so happy to get another opportunity to do so. We can go to the next slide. So in a way, this is kind of my disclosure slide. I just like to give a plug for the Center for Mendelian Genomics whenever I talk about anything, and that we're an NIH-funded center to exome and genome sequence, mostly exome, cases that are undiagnosed to try to discover new disease genes, and we're always looking for new collaborators. So just putting that out there, and the rest of the time will be devoted to reference databases. Next slide. So I'm going to talk actually mostly about Nomad, but I'm going to take you, there's about 7 or 8 reference databases available now that I've used to some extent. So I'm going to describe each one. There are a few others out there that I've heard of but actually have never used, so I haven't included those. And then I'm mostly going to talk about Nomad, telling you sort of where the populations come from, how to look up a variant, check the read support, if a variant's absent, how to figure out if that region is covered. I'm also going to go into constraint metrics. And then I was looking in the email and filtering allele frequencies were mentioned, but it's a pretty broad, it's a pretty heavy topic. So I actually have 20 slides on it, but I put them after everything because I don't think we'll have time on this call, and I'd recommend having James Ware or Nikki Whiffen give a dedicated talk on that. And if there's a little more time, we could talk about GTEx, the RNA expression database, if people are interested in that, or there's lots of other speakers who could address that also. Next slide. So you all are aware of this, but sorry, I have a lot of animations in my slide, so you can actually just flip through to show these. But when we look at an exome, we see many rare variants. So we have about 500 rare missense variants, about a third of by in silico predictors thought to be damaging, 100 loss of function variants, many of which are common, but there are some that are rare, about 100 rare variants in known disease genes. If you use something like the Human Gene Mutation Database, you have 50 reported disease-causing mutations, and everyone has 1 to 2 de novo coding mutations. So there's a lot of variants to look at in every exome, and so there's a lot to make sense of, and that's why we use reference population databases. So the next slide shows just that plus there's a bunch of sequencing errors. We compare basically one child to the general population to try to make sense of their rare variants. First off, to figure out which variants are rare, and then to make sense of them. So next slide. And when we do this with any large population database, this just shows you for ExAC compared to a much smaller, an order of magnitude smaller database called the Exome Sequencing Project, which was one of the first population databases. We can take the number of very rare variants with a 0.1% allele frequency down from about 1,000 when we used a smaller population database to less than 200 variants that we have to look through for very rare variants when we use something like ExAC, and a little bit further when we use Nomad. So that's a 5-fold reduction, so that's been very helpful in letting us know where to focus our energies. Next slide. So these are the main reference population databases that are out there. They're ordered in size, not as opposed to like age of them being available. And I have a slide on each one, just going through from left to right. So the next slide shows you the Greater Middle Eastern database. Oh, sorry. Yeah, that's fine. Which contains exomes from individuals in the Middle East, but it also includes people who have Mendelian disease. But the thought is it's a mixture of a bunch of different sites in the Middle East. The Middle East, you'll see that is the bright green stripe on the bar graph there, and we don't really have good representation in any of the other databases of this population, so it's one of the only places we can look up these population allele frequencies. So it's just helpful to know about, but we don't. So we use it when we have a patient from a Middle Eastern background. The next slide shows 1,000 genomes. So it's one of the very first databases that was available. It has exome and low coverage genomes from many diverse ancestries, and because of its ancestral diversity, we still use it when filtering our exomes for analysis. And the next slide shows the Exome Sequencing Project. This is one of the first dedicated, the second dedicated sequencing project to have reference populations. It's mainly European and African. They are from, people talk about them being from healthy populations, but they're actually just from the general population like Nomad and ExAC are, and that they were recruiting out of cardiac clinics and other clinics, so they're not necessarily like a robust, healthy cohort in any sense. We actually don't use this in our filtering because most of the samples, or a large proportion of the samples from ESP are included in ExAC, and so you'd be double counting if you were to use both databases. Next slide. Oh, apparently these are out of order, sorry. So Discover EHR is from Geisinger Regeneron. They share, it's publicly available, but they've binned the rare variants. So anything less than 0.1% allele frequency is just binned, and you can't tell how rare something is. And a caveat is greater than 25%. And as they expand in their Western Pennsylvania population, probably even more of their cohort is related. And so it's not a great reference population database for that, but it's still a very interesting database. Next slide. So OpenSearch is the Human Longevity Incorporated database. They do the same thing where they bin rare variants. You can't download their data set, but you can load and upload a VCF of your own for annotation in case people are interested, and you can go to the website and see a table with all the variants. Next slide. So ExAC is one of the first aggregated, so we didn't do any additional sequencing for this data. We just aggregated existing data through access through dbGaP and through researchers at the Broad who were interested in sharing their data. It has good representation from 5 different ancestries and really changed the field when it was released in October 2014 and became the standard for reference databases for molecular diagnostic labs. Next slide. Bravo is the other database that we use actually a lot. It is a whole genome database, so it's the largest whole genome database right now. They are unable to, due to some restrictions on the data, provide any ancestry information, and you can't download the complete data set, but you can look up variants on their website, which is based off the ExAC site too. They have not removed related individuals in their data set, but there isn't the same kind of enrichment you would see in Discover EHR, but they could exist. And the other caveat here is their data is all mapped to HG38, whereas I think all of the others are mapped to HG37. Yeah. So next slide is Nomad, and then we'll go to the next slide where it goes through more detail about Nomad. So data is provided from 107 different PIs from over 138,000 individuals, which is 123,000 exomes and 15,000 whole genomes. It's all Illumina data. It's all processed through the same pipeline. So when we have external Illumina data, we bring it in and we process it through GATK through the same pipeline that we use on internal data. We've called all the data in one giant call set, so it's just the most uniform you can get for one of these for this data, although there are different platforms of exomes in the data set. We have a sites VCF of the entire data set that has the not individual level data, but the data for each variant that you can download. It's huge now, but you technically could download it, or otherwise you can access it and annotate through your pipelines with it. It has all the allele frequencies and the population allele frequencies. There's not consent to share individual level data, and we do not have phenotype data available on very many of the individuals. Next slide. It includes cases and controls from common disease studies. There's no Mendelian disease studies knowingly included. I'll go a little bit more into the phenotype so you can, there's some debate about what you can consider Mendelian. And then in Nomad, we also have Ashkenazi Jewish samples that have broken out as a separate subpopulation. We have something called PopMax, which reports the population with the highest allele frequency for each variant. And in that, we, in general, exclude Ashkenazi Jewish and Finnish samples because of their founder effects from our PopMax calculation. So we just look at how common the most common the variant is in European, Latino, African, South Asian, and East Asian ancestry groups. Next slide. It's about half male, about 55% male, and the average age is about 55. So these are mostly adults in the database. There are a few cohorts that do include some individuals under age 18, though, but it's a small minority of the data set. Ancestry and sex are inferred from principal component analysis and from the genomic data rather than being reported. So they're, we think, more accurate that way. So this is the example of what this is the PCA for the Nomad data set here. So you can see in orange is the large European group. Coming off the orange in the different colored blue are the Finnish and the Ashkenazi Jewish. Up on the bottom in maroon is the Latino. The East Asian's kind of in the middle there in purple. The top one is the South Asian, and then the African are now coming around in black. So we're able to discretely separate these populations out, but you'll see there is admixture between the groups, and for the most part, samples get assigned to whichever one they're closer to, although there are, at a few percentage, I don't know the number, of the samples are considered other that we're unable to classify into a population. Next slide. So these are all the cohorts that are included. You can see these on the website. On the next slide, I have grouped them into the general phenotypes that for the most part are what is included. So there are a lot of population studies, so samples from 1,000 genomes and exome sequencing projects. There's some twin cohorts in here where only one of the twins would be included. We've removed first and second degree relatives, but not further than that. There are atrial fibrillation and myocardial infarction cohorts. There aren't any cardiomyopathy cohorts as far as we're aware. There are there's tons of people with type 2 diabetes in these data sets. There are people with bipolar, schizophrenia, and migraines. Sometimes people feel that schizophrenia could be a Mendelian disorder. In general, we haven't found, Mark Daly has done a lot of work there and hasn't found a lot of signal there. I will say one of the most common inquiries we get is my variant is involved in neurodevelopmental disease, and I see it in 10 people in Nomad. So I think those must be all people with schizophrenia, and that's why you're seeing it in the population database. And in all of the inquiries I've gotten like that, it has never been the case that they are enriched in schizophrenia. That's not to say that it can't happen, but just it is not something we have seen being a problem. There's Alzheimer's, there's ALS. There obviously are some Mendelian forms of ALS, so I actually think that's a little bit of an issue. There's inflammatory bowel disease, also some Mendelian-ish components there, I guess, but not straightforward Mendelian disease. There are people who have cancer, so there are TCGA samples, but they're the germline samples, not the cancer samples. And we have in the past released some subsets, and we're working on some new subsets from the Nomad data set to release. So there will be a subset that is what we call cancer-free, although many people in the general population will get cancer, so it's not that the people in this cohort don't have cancer, it's just that we don't have any knowledge of them having cancer. There will be a subset that is not from a psych or neuro cohort, and then a subset, I think there's a planned subset that excludes diabetes samples. I'm not positive about that, but I think that's in the plans. None of those are, we're still working on the final Nomad release, which I'll get to in a future slide, so none of those are available yet. Next slide. Next slide. So I just wanted to go through how to look up a variant in Nomad. I'm assuming for this audience that many of you, or most of you, or possibly all of you know how to do this already, but this is the general example that I use. So in cystic fibrosis, you need 2 mutations in trans to have the disease, and so we can see people with 1 mutation or 1 variant, that's 1 pathogenic variant for this disorder in the general population who are carriers for the condition. So if we go to the next slide, this is how you look up a gene. You go to the nomad.broadinstitute.org, or I really like the new Nomad beta site, and so I recommend that. I think that most of my slides are from the beta site that we'll be seeing here. So if you type in your gene of interest in the search bar and hit enter, and go to the next slide. This is the beta site where you'll see a lot of information on this page. So at the top, you have the name of the gene. Underneath that, you have some transcript information, a link out to UCSC, a link to OMIM. So the blue mountains and the green in between, or the green across, is the coverage. So on the left-hand side, you can see the X coverage. So some of the exons we have up to 90X coverage. Some of them are smaller and only at 40 or 50X coverage. This is the average coverage of each exon in blue. And then the genome data, the average coverage is in green. You'll see it's about 30X across, and it tends to be pretty smooth. We don't have very much of the introns represented here. Below that, you'll see the canonical transcript. And one thing to note is on the left-hand side, you'll see a little arrow that either points to the right or to the left, depending if it's a plus or a minus strand gene. And then below, you'll see all the different transcripts from ensemble transcripts. We use GenCode. Of note, we are using a slightly outdated version of GenCode, and that is because as the new versions of GenCode have come out, they have switched a lot of their annotation to really supporting HG38 rather than HG37, and so some of the tools we use aren't really available for the newer versions of GenCode. And so we're working In the future, the next version of Nomad will probably be on HG38, and so we'll be able to update our gen code at that point. Below that is the variants, so you can see them all kind of squished together there in a line. The height is related to how common the variant is, and the colors gray is synonymous, yellow orangish is missense, and red is loss of function. Sorry, I think green is synonymous. I think gray is non-coding. And then at the bottom, there's a list of all the variants in the gene, and you can click on a link to any single one of those. The other thing you can do on this page is you can include filtered variants. So the whole VCF is available to look at on the website, but only the variants that are passing the filter are showed on the default view. And then the other thing you can do here is right above the variant list also, there's something that says Nomad in the drop-down menu. You can choose to look at ExAC, only variants instead, so that's all adjustable there. There's a search bar, so if you click the next slide, you'll see that I want to look for the most common variant in CFTR, the phenylalanine 508 del variant. So I can type in whatever I want there, and it brings up just those variants. I like that it shows where the variant is, and so you can see which exons it's falling in, which is nice when it falls in exons that are only in some of the transcripts, so that can be handy. And then I can click on the link to go to the variant page for that variant. So if you click the next slide, and then again. So this is what the variant page looks like. The box in the orange at the top has the allele counts in exons and genomes. In general, we don't see a huge difference between exons and genomes. There are some subtleties there. Obviously, things that are non-coding may only be in the genomes because we might not be capturing them in the exomes. Things that are only captured in a small subset of exon captures, you might see like low coverage in the exomes, but still some there. I will say the ancestry breakdown of the samples we have genomes from is not as diverse as the exome samples, and so you will sometimes see that affecting the allele frequencies, and so there can be some discrepancies between the two when the exome samples are more similar size to the genome samples. But in general, this is the variant page, so if you click the next slide, and then click again, we're going to zoom in on this variant information. So these are all the populations here, the allele counts for this specific variant. And then I've done some math on the side where this is the most common carrier variant, the most common variant in cystic fibrosis. And the European carrier frequency just from databases is known to be 1 in 41. For this specific variant, it's more like 1 in 25 for Europeans for cystic fibrosis, but 1 in 41 for this variant. And so I took the total number of people who were genotyped in the Europeans at this site, which is 126,000 divided by 2, which is about 63,000, times 1 over 41 people should be carriers. And so I expect in this European population to see about 1,500 carriers, and we see 1,531. So we're getting a nice representation of expectation for the carrier population. And I think that's one of the really nice things about having a data set that has so many different cohorts in it, is you do get kind of an averaging out of any biases in any single cohort. Next slide. Next slide. Oh, actually, can you go one back? So I do like to point out that while this is a very common variant in Europeans, it's actually not seen in any of the 9,000 East Asians in this population. So it just shows you the differences in ancestries. Next slide. You want to look at the read data supporting any variant, and it's present at the bottom of each page. If you go to the next slide again. Sorry, those are kind of out of order also. But anyway, so one of the other things we're interested in is how many homozygotes you would expect to see for this variant. I would actually predict that there would not be anyone with cystic fibrosis in this database because it's supposed to be depleted for severe pediatric Mendelian disease. You would expect to see 9 homozygotes just by doing the math there of how many carriers we see, but we actually only see 1. To me, it was surprising we saw 1 at all. But overall, it just shows, it highlights that there can be people with disease or with diseased phenotypes in Nomad because of a variety of reasons. That could include sample mix-up when a sample goes into a cohort. It can include somebody participating in a study that is selecting for some trait that has nothing to do with their Mendelian disease and wouldn't exclude someone with Mendelian disease, or it could be that these people are non-penetrant for their condition. So just to keep in mind that occasionally we do see some disease variants, and there's lots of explanations for why those are present. Next slide. One of the important things is this homozygous variant real, though? So we do go to the data here. And so you can see that it's pretty clean. I will say most of the reads are blue, and so that's because they're on one strand. And so this initially looked like there was strand bias, and I worried about that a little bit. I'm not showing you here, but if you were able to scroll down, you would actually see there's lots of pink reads below. So I think this variant looks fine and looks real. So next slide. This always raises the question of how do I tell like what variants aren't real, like what should I be looking for? And so I'm going to take you through a number of examples of this. So we can go to the next slide. So the first is things that are called by algorithms or by VEP to be loss of function or protein truncating variants, but we have for some reason think they're low confidence loss of function variants. This can happen for a number of reasons. So it could be a dubious transcript annotation in that it might be a nonsense variant in an exon, but when you look at the exon, it's really poorly conserved and it's not included in very many of the transcripts. And so maybe it's not like a very important exon or even a real exon. So those get flagged by Lofty as a tool that flags those, and we mark those in the browser. And the other is protein truncating variants in the last 50 base pairs of the penultimate exon or in the last exon wouldn't be expected to trigger nonsense-mediated decay, so we mark those as low confidence loss of function. That's not to say you can't have pathogenic variants there, but just that they wouldn't necessarily cause loss of function. You should look at them more carefully. So those are flagged, not necessarily artifacts, but just flagged variants. Next slide. Sometimes you look at a variant and what you see is a region like this, where you see a large number of variants. So you have a few synonymous variants in green across on the left. You have kind of smattering of variants that only seem in like one or two of the samples and aren't very recurrent. You have an insertion, that's what the purple bar is on the right, and you have a deletion in one base pair in the middle there, that's the black horizontal bar. So one of the other things you'll notice is that it's calling 5 variants at the top there, not counting the insertion, that's most likely calling also, but and they have different allele balances, so like something funky is going on here. And if you look at the sequence at the top, you'll notice that there's a lot of Cs, and so I think this is just a region that's hard to sequence. This gets actually flagged as a low complexity region, and there just is a lot of both slippage and mapping issues here. And so if you go to the next slide, if you looked at this variant in the variant list, we have actually flagged this as a low complexity repeat region, and it should be interpreted with caution. But in general, anything where I see a bunch of variants in a read makes me very worried. Obviously, there are haplotypes, and you can have 2 variants or 3 variants on the same haplotype, but you should see them then consistently on the same reads. Next slide. Homopolymers are a place where there's an enrichment for artifacts from PCR. Unfortunately, it's also a place where you are more likely to get slippage from the polymerase in vivo also, so that you're enriched for true variants also, but it's just important to be aware when you see something in a homopolymer run that it could be a PCR artifact from sequencing, or it could be something that's real, and so you need to be saying or confirming any of these or thinking about them with more caution. Next slide. Multinucleotide variants are really interesting. So that's when you have 2 variants within the same codon in cis, as you see here. When you interpret each one individually, so the first variant here is a T to a C, so the red T to a blue C. It's a serine to proline missense variant. The second one would be interpreted separately as a serine to nonsense variant. But when you interpret them together in cis, as you should, because they're within the same codon in cis, then you get a serine to glutamine variant with missense variant, which is an entirely different missense variant. So there's nothing, the VCFs, all the pipelines I'm aware of call these separately and don't put them together properly, but they're obviously like very easy to spot when you look at the read data. And so we find all the time When we see protein truncating variants in genes that shouldn't tolerate protein truncating variants, they are enriched for these multinucleotide variants. So it's just an important thing to look for. We have them flagged in ExAC, but we haven't flagged them yet in Nomad. We're working on that. You can see the same kind of situation with a complex indel where you might have a two base pair deletion, and then not necessarily within the same codon, but a little bit downstream, you have a two base pair insertion that restores the frame. So another thing to look for. Next slide. And then you can see skewed allele balance, so this is only present in about 20% of the reads here. I think that if you go to the next slide, I think I've annotated that. And so you just should make note of these. These could be somatic mutations. These could be PCR artifacts. There's lots of explanations for them. We have not flagged these. In ExAC, these were coming up a lot more because they're actually part of the training data and part of the QC in Nomad. So we have filtered more of these out in Nomad. Next slide. You can just flip through this. Sorry. So how to determine if a variant is really absent from Nomad? Because often you go and you look at your variant of interest, it's not in Nomad, and you're like, great, I'm going to call that absent. But there's a few things you have to be aware of. It could be absent from Nomad because you're looking at the position you're looking at isn't the same position in the canonical transcript that we're showing on Nomad. So it's important to make sure you're looking at the right thing, and chromosome coordinates are often the best way to do that. Second, the position is not covered in Nomad, or third, what you're interested in is the variant is just not in Nomad. Next slide. So you can type any variant position in. I recommend not typing the whole variant in, but just typing in the chromosome, colon, and then the position. If you were to type like a C to A variant, I feel like that messes it up sometimes, and it's better to just type the chromosome coordinate. Nomad is not enabled for HGVS searching like that, although we've talked about trying to get that incorporated in the future. So when you look at variants, I'm looking for this variant in orange at the left, this proline 273 threonine. It's not in Nomad when I first looked. I clicked filtered to include filtered variants, and this variant is not there, but I do see another variant of this position. That's a synonymous variant, and what's nice about that is it tells me that the allele number, or number of people genotyped at this site, is number of chromosomes genotyped at this site is 65,000. So 65,000 divided by 2 gives me about 32,000 people genotyped at this site. So it's not totally fair to say it's absent from all of Nomad, and that implies it's absent in 135,000 people, but it is absent in 32,000 people, which is still a pretty good control population to look for it in. Next slide. Okay, I get a lot of questions about what are the differences between Nomad and ExAC, and should we be using ExAC or should we be using Nomad? In general, most of the samples that are in ExAC are in Nomad. There are a few that were excluded for two kind of main reasons. One is sometimes the BAM file just wasn't available when we did the second call of Nomad, and sometimes a sample was included in the call set but got filtered at the sample QC step because that was it was a new QC that was done on Nomad. So there's some differences in sample filtration there. But overall, most of the samples in ExAC are in Nomad. So seeing something in Nomad and in ExAC is double counting. You shouldn't be including those as separate interpretations. Overall, the QC process has matured a lot. We've learned a lot of things from building and analyzing ExAC that we have applied in Nomad. Additionally, Nomad has genomes and exomes, and that's added a lot of complexity to the QC process. In ExAC, it was mostly VQSR based, which everyone uses through GATK, and there are blogs about that if people want to know more about that. Now we're using a random forest model that's trained on a number of different factors, including the type of variant, which I think I mostly mean single nucleotide variant versus indel here, a bunch of quality metrics, the depth of coverage, the allele balance of the variant, the position in the read, the strand bias, and some other more nuanced parameters. These are the main things. In general, the current Nomad release is still beta. We are actively working on the next version, which we hope will be the final call set for this version of Nomad. We don't expect very much to change in terms of specific variants. I think there might be a few new samples added that we have gotten permission to share since the last release. But what will change is where the pass-fail line is drawn for filtering, as we've still been working, like a large team, mostly led by Grace Chow, Laurent Francioli, and Konrad Karczewski have been working on this QC for over a year now. And we think we've got a little bit of a more robust way of doing it. In general, right now the filtering is a little more conservative in Nomad in that versus ExAC, and that I think there are more real variants that are being filtered out. And so I think that that line is going to move a little more in including a few more variants into Nomad. We're hoping to have the final release later this year. In general, we are using internally Nomad for all of our allele filtering for exome and genome analysis. But if the variant is absent in Nomad, we do kind of look and see if it is present in ExAC. It's less important for something recessive, but if you were looking and thinking about something de novo dominant, we would still check ExAC. Next slide. Constraint. Okay. So this is work by Caitlin Samoha, who was a graduate student in Mark Daly's lab, who now has moved over to Matt Hurl's lab at the UK at Sanger. So and Konrad Karczewski has taken over this project and is working on the new version of Constraint for Nomad, which is not available yet. I think will be part of the next QC release, but I'm not entirely positive. Next slide. So hopefully most people have seen this before, but just to review, in general, mutations will arise across the genome and exome at pretty even rates, so they should hit genes fairly evenly. Obviously, CpG sites, particularly methylated ones, are more mutagenic, so there are some subtle differences. But in general, next slide, you'll see mutations fall across the genome. What happens is not large differences in mutation rate, it's large differences in what variants are passed on. So genes that are tolerant of a lot of variation, those variants will be passed on to the next generation because they don't do any harm. Whereas genes that are constrained and intolerant of variation, those variants will not be passed on to the next generation. So if we go to the next slide, and again, so what we're looking at is the population today. Sorry, can you go back one? And you'll just see that in tolerant genes, you'll see a lot of variation building up, and in constrained genes, you'll see very little variation. So in the next slide, so we've developed, basically, Caitlin's built a mutational model that I'm not going to go into details of, but it basically counts up how many variants you expect to see of synonymous missense and loss of function type in the gene, which takes into account the sequence context of the gene, the size of the gene based on the sequence context, and also looks at regional divergence differences between humans and macaques. So for every gene, we have calculated this, and for most of them, this information is publicly shared. The genes that this information is not available for are genes where the model isn't behaving well. So there are some genes that are outliers for synonymous variants, in which case we don't think these are reliable metrics. The other caveat is if a gene isn't included in the version of GenCode that we're using, then we don't do the calculations and annotations on it. But otherwise, most things are here. So here for this gene, we expect 235 synonymous variants. We see 200. So that is about what is expected. For missense, we expect 478. You see 297. So that is fewer than expected, and is actually significantly fewer than expected when you see that the z-score, or it's 4 standard deviations from the mean of expectation. And for loss of function or protein truncating variants, we expect 45, and we only see 1. And so this is also very depleted. And instead of doing z-scores for the PLI, because we expect to see fewer loss of function variants in general, z-scores were somewhat correlated with the length of a gene. And so Caitlin used what's called an expectation maximization model, where she basically just said, do things look more like they're haploinsufficient and that where you have 10% or less of the loss function as expected? Do they look more recessive, where you see 50% as expected, or do they look more completely tolerant, where you see no depletion of loss of function? But in general, this is a tool for really pulling out haploinsufficient genes. And so a PLI greater than 0.9 is what we think of as a score that implies a gene is haploinsufficient. So 1 is very high. Next slide. So this gene, JAG1, is associated with Alagille syndrome, which is a pediatric onset cardiac and liver disease. So you would expect it to be it is a known pediatric disease gene, so we would expect it to be haploinsufficient. We know it is haploinsufficient, so that score is working. One caveat here is this model only really counts nonsense and essential splice disrupting protein truncating variants. It does not take into account frame shift and indels, which you'll see if you go to the next slide. I think I put a giant X over that. And that is because it is very hard to know what the expectation is for indels, since an infinite number can occur at any position in the genome, and we don't really have a good way to predict if there's certain sites that are more likely to occur or not. So people are working on this, but an expectation model for indels has been challenging. Next slide. So PLI scores, I'm sorry, this one's animated again. You can flip through these. So haploinsufficiency is when having only one copy is insufficient to maintain a wild-type phenotype. PLI is the probability of being loss function intolerant, and a high PLI is greater than 0.9. In general, this works well. It pulls out known haploinsufficient genes. The more of them have a high PLI score if they have a severe phenotype versus more mild phenotype. In general, genes that are recessive have a low PLI. Next slide. So we have 3,200 haploinsufficient genes, and greater than 70% of them at the time of the ExAC publication 2 years ago had not been associated with disease in OMIM. So there's a lot of sort of candidate disease genes that have been highlighted by this tool, and really shows how we're learning new biology from these reference population data sets. Next slide. I want to highlight this caveat that for adult onset conditions, where the age of onset of the condition is after, for the most part, fertility, you do not see an evolutionary signature the same way. And so, for example, for BRCA1, we know it's a haploinsufficient gene that increases the risk of breast and ovarian cancer, but many people with pathogenic variants in BRCA1 still reproduce, and so we do see these variants being passed on, and we do see them in reference population databases, and so you do not see a high PLI score here. So that's just an important caveat to think about as you are analyzing your genes. It depends what your phenotype is and the age of onset. Next slide. And then again, CSTR, the cystic fibrosis gene, recessive genes, we expect to see loss function mutations in the general population because they'll be carriers and they should have a low or just mildly elevated PLI. And so it's not a good tool for looking at recessive conditions. Next slide. I talked about missense, but there's 1,800 missense constrained genes that have been identified, and then regional missense constraint I'm going to briefly talk about on the next slide. So all the constraint data is available on the browser. It's available either on the ExAC browser or on the Nomad beta browser. When you go to that site, the table will be blank in the top right-hand corner, but if you click the ExAC part in the red box inside the larger red box I have there, that switches over to only viewing the ExAC data, and you'll see the constraint data there. And once the Nomad constraint data is available, those tables will be filled, so you'll be able to see all of that data there. Next slide. We don't have regional missense. So Caitlin's basically applied the same thing to saying, when you see genes that are depleted from missense constraint, is it in the entire gene or is it only in a part of the gene? She does it by sort of dividing the gene up, and then once she figures out which exons are the most constrained, kind of bleeding into the exons and seeing where the boundaries of this are. This is not on the ExAC browser yet, but this is a prototype that we've been working on. So we're hoping to show the observed over expected depletion for different regions in the bar like this. So you should see this coming soon. If you want access to it now, it's available on the Decipher site. So next slide. If you go to the protein view for any gene, I personally love this view on the Decipher site where you can see all the ClinVar and Decipher variants and the domains, but we have modeled ours similarly for the regional missense constraint so that people will not be confused when looking between the two sites. So this is the same gene here, just showing those regions of missense constraint. So that's already available. Next slide. So in conclusion, I think reference databases have been really critical for us to figure out which variants are real and to help us interpret them. I think there's a lot of extra things we can use by looking at the reviewing the read data and the coverage to really see if a variant is there and if it looks real when it is in these databases. Next slide. Sorry, next. Constraint, I think, has been very powerful. We often are able to be much more interested in de novo variants and other variants we find that are rare and in constrained genes. And I think a lot of the new genes that are being described recently have been constrained genes. And so this is a very powerful thing to look at. It's also very, I think the regional constraint is going to be very interesting in finding these regions as one of the ACMG criteria that are depleted for rare missense variation. And then we're working on the next version of Nomad now, which we hope will have about 70,000 genomes. And we're aiming for, sometimes they say end of this year, but I think early next year will be more realistic when that will come out. But that will be on HE38. And then I think the next slide just shows the team. So this is most of the team, and there's other people also who work on the Nomad website and the resource. It's obviously a ton of work that's led by Daniel MacArthur. Conrad Karchevsky, Laurent Francioli, and Grace Chow have been really hip deep in variant QC of the data set for the last over a year, and Conrad's been working on it since ExAC. So it takes a lot of people to really bring this data to you. Jessica Elfoldi is the one who gets permission to use all of the data, and Monko Lek built the original and built and QC'd the original ExAC data set. So that's what I wanted to talk about today. Happy to take questions. Are there any questions I can answer about the data set?
I had one question. If you could comment on the multinucleotide variant, you mentioned that you might one of those variants, if looked at separately, might be counted as nonsense or loss of function. Is that corrected for with the constraint score, or is that something we need to be wary of as well?
It's definitely not taken into account for the constraint score. I mean, all the pipelines are interpreting these as two separate variants. It's not adding. I mean, it's a minority of the variants in terms of thinking about constraint. So it's not going to change the numbers for constraint very much. But for example, when we have a gene that we know is a haploinsufficient gene that's well associated with a well-described syndrome, when you look at a lot of, and you still see some loss of function variants in Nomad, most of those are not really true loss of function variants. And if there were 10 of them, sometimes like 2 of them will be multinucleotide variants. But we can normally account for generally about 70 to 80% for why they're not really resulting in loss of function.
Okay, thank you very much. This was awesome.
Hey, Anne, it's Jessie. I had a question. The subgroup thing sounds really, really interesting. I was wondering if you guys have this information, if it would be possible to also, I don't know, make subgroups in case people want to kick out anybody who's, under age 30 or under age 40.
Yeah, I can pass that on to the team to see if they'd be willing to do something like that. We'll put that on the request list. Other questions or suggestions?
This is Jenny. I have a quick question. So when you're thinking about recessive disorders and you're thinking about the expected number of homozygotes, so as you showed for your cystic fibrosis example, there was only one homozygote. You were even surprised to see that. But is there any plan to include any statistics on that type of data for ExAC? So for specific variants where it might be causing a recessive disorder and you're seeing fewer homozygotes than you would expect if it was not disease causing. Does that make sense?
Yeah. So for all of ExAC or all of Nomad, it doesn't work because of the different ancestry groups. You can do it within an ancestry group. But even then, there can be some nuances to it if a carrier variant is more common in Italians versus some other group. And So in general, if you try to look at things that violate like Hardy-Weinberg expectations, it's just a mess in ExAC and Nomad. What it mostly pulls out is mapping artifacts or some other kind of artifacts in the data, which, because their artifacts don't follow Hardy-Weinberg. So it seems like a thing, and people keep trying to do it, but it's just not working to pull out any kind of interesting biology thus far.
Okay, that's good to know. Thanks.
One of the other things we're working on is how often you expect to see 2 rare missense and/or loss of function variants in a specific gene in the general population, so that you could say, I'm interested, I have this candidate gene, I see 2 rare missense variants. Is that unusual? Because if you had 2 rare missense variants in Titan, we actually see that present in 40% of people in Titan, and so obviously that would not be interesting on that basis alone. So for each gene, we're trying to calculate that using the individual level data and then be able to share kind of a number for each gene. But that's also been a work in progress.
That's fantastic. That's great. And presumably with these genomes, I mean, you wouldn't know for that type of data if those variants are in cis or trans, but just knowing statistically that seeing 2 variants in that gene together is less common than you might expect, would suggest it's a disease-causing gene.
So Laurent Francioli has been working on statistical phasing, because we don't have trios for most of Nomad. So he's been working on statistical phasing of 2 rare variants in Nomad, and he has a model. He's talking at ESHG next week for if anyone's going to be there, or this week. I don't actually know when it is, but he just gave a practice talk. It was great. And so he will be talking about how he can predict with about 80% confidence if a variant is in cis or in trans within a gene in Nomad. It works for very rare variants. It doesn't work if very well if one of the variants is absent from Nomad, but otherwise, if they're both in Nomad, we have some ability to estimate if they're in cis or trans based on how often we see them together in other people in the population. So that's also, once he's finished that work, that will also be released in some way on the browser.
Great.